The solution to the problem is the genetic stability, because with this high replication rate of microbial cells, you always have this genetic drift. And this is already pronounced in a simple fat batch process. But when you start to do that in a continuous manner, the process has to be terminated within a few days. Because if you use a classic cell line, so the cells will mutate so quickly and there will be no productivity within 4 to 5 days.
What if the biggest bottleneck in bioprocessing isn't your downstream equipment or your facility design, but the production host itself? Today, I'm joined by Juergen Mairhofer, who is the CEO of enGenes Biotech, who has spent over a decade engineering smarter microbial platforms and pioneering continuous manufacturing using microbial cells for recombinant proteins and plasmid DNA. In this conversation, we dig into what it actually takes to move from batch thinking to continuous reality. Let's dive in. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Juergen, to the Smart Biotech Scientist. It's good to have you on today.
Great, David, to be with you today, and I'm really looking forward to this very nice conversation.
Juergen, share something that you believe about bioprocess development that most people disagree with.
That most people disagree with. I don't want to be provocative here, but I think that the commodity CDMO model rewards utilization over innovation, and that's why it's becoming unsustainable. So what we know is that CDMOs optimize for utilization of their existing steel tanks and not really for the productivity of the process. I think the problem that we are seeing in the industry and the traditional business model they are driving rewards risk minimization and capital utilization on their end, and they are not really willing to innovate. When you make money by selling fermentation time, you have zero incentive to make processes faster and more efficient. I think this is the core problem or the root cause of the problem. And when you look into documents like recently read one from Roland Berger, and the data in there shows that the CDMOs with the right business model really focused on innovation can avoid significant price pressure. This is exactly the point. So the commodity model is under severe pressure, while innovation-focused players thrive. And we as enGenes Biotech, so we deliberately didn't build enGenes as a traditional CDMO. So our model is based on real process development using proprietary technologies and then out-licensing these technologies. So enGenes makes money when the process gets better, not when it gets longer. So this is the basic principle. And this aligns our interest with those of our customers. So we are the innovation partner, let's say it that way, that CDMO and pharma companies are in need of in these difficult times.
Before we dive a bit further into your CDMO model and what exactly you're doing in process development, draw us into your story, Juergen. What sparked your interest in biotechnology, and what were some pivotal moments leading to your current CEO role?
So what are the pivotal moments? I think the pivotal moment was when I started my master thesis here at the University of Natural Resources and Life Sciences, where I did host cell engineering, so classical molecular biology, and where for the first time in my life, I could be creative in a way without other people judging on my creativity. Because if you do painting or something like that, everyone can say, okay, this doesn't look like a dog. But if you pipette small volumes in an Eppendorf tube and you have to wait for 4 weeks until you get a data set out, in between, nobody can judge on if the creative process is good, bad, or whatever. So with my supervisor at that time, I learned that creativity is something really amazing. And being raised in a home where everything was macro, because my father is a car mechanic, where things were judged immediately because you can just do things right or you can do things wrong. But with molecular biology, you have to try and you have to learn if things work out. And this is where the creative process started. Being at that time, I think 24, I think the first time in my life I learned what creativity can really bring on the table. And there was no judgment in academia. So we were able to try things out and see if things worked out. And we were following a lot of weird ideas at that time point, because this was the time when synthetic biology was born, more or less. So starting 2002, I think, with the first publications. And I ended up in a very dynamic field, and I really appreciated that. So being able to be creative, I think this was the spark. And then having the opportunity to do a PhD together with a big pharma company. So my PhD project was funded by Boehringer Ingelheim at that time, and getting also an idea how the big industry works, and that then being able to do 2 more additional postdoc stations with, for example, the Austrian Center of Industrial Biotechnology, so ACIP. I also got more and more insights in how the industry works. And at the end of the day, I realized that the big pharma industry is not the place where I want to grow old. So I come from a sort of a DIY perspective. So I grew up on the countryside in the 90s where nothing was going on. So it was very boring. When we want to have fun, we have to build our stuff on our own. And I think with that mentality, I was able to have the mindset to start thinking about founding my own company. And I think that's the thing where everything started. So being passionate about being creative, building stuff on my own, and then also having the guts and coming from a hard work mentality to be able to really dig into a topic and make it work. So I think this is what got me into funding my own company.
Yeah, I love the way you're looking at pharma, looking at creativity where a lot of people see regulatory constraints. It's a pretty good way to look at it. And then on top, your entrepreneurial mindset and also hard work. And I think that's definitely a sweet spot. So tell us a bit more about what you're doing now with your company, how you're translating scientific innovation into scalable bioprocesses and more.
Yeah, so as I started to explain, I was always working at the intersection of molecular biology and bioprocess engineering. So this was quite new in the early 2000s. So it's the field of synthetic biology coming up. And at that time, people were used to working in silos. So there was like people doing molecular biology, and then there were the bioprocessing people. And within the bioprocessing community, there were the upstream people and the downstream people. And none of them were able to communicate with each other because they had different mindset, they had a different language they were speaking. And the funny thing was that within my PhD project, it was located directly at that intersection. So meaning we had to engineer a strain, so to build a new E. coli strain with a lot of genetic features to be able to do certain tasks. And I brought that strain to another working group at the university doing upstream processing. And I think I was the first guy who was a molecular biologist and then were running fermenters at that time. So that was quite weird. I had to really work hard so that these people taking me serious. But at some point, they started to understood that this knowledge has a lot of value because it's like an iterative cycle. This is what I implemented, I think, here at the university, saying, okay, we have to engineer the strain to meet the requirements of a bioprocess, and then we have to look how the strain behaves in a real bioreactor and not in a shaker flask. And then once we have learned that, we have to get back and improve the strain, and we have to run this cycle iteratively for several times until we have an output that meets our requirements. So this is how I learned to work with the tools I had at that time. And this was fundamental, I think, to founding enGenes Biotech, because all the work we are doing is based on proprietary E. coli host strain that I invented together with colleagues. So what we are capable of doing is we can decouple protein production from cell growth. So at a dedicated time point in the bioprocess, we can stop cell division, at the same time switch on the protein production process that is using an orthogonal transcription system. So in that case, T7 RNA polymerase that is then massively overtranscribing gene of interest. And by having this non-dividing cell population, we can fill the cells with recombinant protein, or we can even secrete the protein to the extracellular space, and at the same time genetically stabilizing the manufacturing system. Because one of the big drawbacks of using bacteria is that they have a very rapid growth rate. So they have a very high turnover. So every 20 minutes, you generate progeny, you have an exponential growth function. And due to that fast growth, the genetics of the cells drift a lot because you always generate variability within your population. And at some point, you end up with a cell that has no longer the plasmid where your gene of interest is located, or has a mutation in an intracellular function. And this cell is then over-proliferating in your bioreactor and stalls your production type. By stopping cell division, you can derail that process. So you're derailing adaptive evolution. And thereby, we were able to really come up with a very stable and robust manufacturing system that we finally commercialized within enGenes Biotech. And this is where the story now, 12 years ago, started.
What kind of products are particularly well suited to produce in your specific strain?
Everything that you can produce using E. coli, so that is non-glycosylated. We are happy to take over this challenge because we believe that the non-growth associated production is most of the time beneficial over the growth associated production, because the growth associated production has these drawbacks that I already addressed, so that the cells try to escape from the burden of producing whatever, let's say recombinant protein, plasmid DNA, small metabolite. It doesn't really matter. They want to escape because their objective function is to generate progeny as fast as possible. And they have absolutely no interest in serving the bioprocess engineer. On the other hand, you have to come up with a smart solution to get the cells to the point where they have to do what you want them to do, and to reduce the degrees of freedom that they have. And by playing this trick that we are playing on the molecular biology level, we block the RNA polymerase of E. coli, so the cell can no longer produce its own messenger RNAs, and therefore it cannot ramp up a stress response, because the stress response is always meant to escape the metabolic burden you exert on the cell by forcing it to overproduce your biomolecule of interest.
Before we continue, biotech founders, I want to talk directly to you. Do you know that feeling when an investor asks about your manufacturing strategy and you freeze? One wrong CMC decision can cost you $2 million and 18 plus months of delays. Yet many CEOs are flying blind on these critical decisions. That's why I created the 1-week CMC strategy assessment. In 1 week, you get a personalized assessment of your molecule and development stage. We identify critical gaps before they become million-dollar mistakes, plus a strategic roadmap for high-stakes investor meetings. One CEO told me, "David took me from feeling powerless about our CMC roadmap to confidently pitching investors. An absolute game changer for time-pressed CEOs." Whether you are preparing for Series A, approaching IND filing, or facing scale-up challenges, this gives you confidence to ace investor and board conversations instead of surviving them. Ready to stop second-guessing your CMC strategy? Check the link in the show notes and secure your spot. Because your breakthrough therapy deserves a solid CMC strategy. Now, let's get back to our episode. And if I may put it simply, basically your cell line gets you a much higher titer, correct? Because you have more energy available for your protein of interest.
Absolutely. So all the metabolic resources are channeled into production of the protein of interest or replication of a plasmid or production of a bioconjugate. And this allows us to come up with very high titer processes, at the same time, very well scalable, because we don't have this process variability that can happen by plasmid loss or accumulating mutations. So also in the scale-up process, the processes we develop are much more robust and stable. And I think this is the big advantage of the technology that we are using.
And what is now the ratio between traditional fed batch processes and continuous in your programs? Because I know you're big on continuous, but what is the current ratio?
I mean, the current ratio, to be honest, is that we have been developing microbial continuous processes, and we are the first movers in that field. So I don't know of any other company that has successfully accomplished running an E. coli process fully continuous for the amount of days that we have achieved. So we are currently talking about 40 days. So I think we are the first mover here. So we're currently trying to scale up our process on our own. So currently it's running at the 1 liter scale, very productive already with a small footprint. But at the end of the day, we are looking to scale that up to 100 liter scale, producing bioreagents. So this is what we are currently looking into. And we are looking for partners who are willing to adapt this process scheme for biopharmaceuticals, for example. So insulin would be an amazing case, for example, because this is something that needs to be produced at an amazing scale. At the same time, the costs have to be low. I mean, what we are struggling with at the end of the day is what you mentioned early on, is that the industry is very risk averse and conservative. For mammalian cell culture, continuous processing takes off at the moment. So we see that there is a lot of interest in that. Just very few companies are able to master that, but some of them are. But for microbial bioprocessing, I think we are just at the beginning. So this is something we have to push through now. And we see that it gets traction, but it needs someone who is willing to adapt it and to further develop it together with us.
What is the reason that E. coli is lagging behind? Because as you said, several companies are mastering continuous processing with CHO, but what is more complex, either on a technical side or perhaps there's some economics on the E. coli that changes that?
The solution to the problem is, as I have explained before, it's the genetic stability, because with this high replication rate of microbial cells, you always have this genetic drift. And this is already pronounced in a simple fed batch process. But when you start to do that in a continuous manner, the process has to be terminated within a few days. Because if you use a classic cell line, so the cells will mutate so quickly and there will be no productivity within 4 to 5 days. That's what we've been seeing by using the classically BL21 strain, for example, in head-to-head comparison with our technology. So this is really pronounced in a continuous mode, and I think the tools haven't been available to tackle that. But now, with our technology, we have a genetically stabilized host cell, and we have a proprietary approach how we do things, because we not run the continuous process just in one vessel, so in one chemostat. We run it in 2 connected chemostats. I mean, this adds a bit of complexity, but at the same time solves the problem with the genetic stability, because in the first vessel, we grow our cells. So there the biomass production is going on. And then we pump our cells into a second vessel where we decouple protein production from cell growth. So we stop cell division in the second vessel, thereby genetically stabilizing the system. And then the production goes on in the second vessel, and we are in fully steady state. So everything that goes in on the one side is equal what goes out on the other side. And by playing that trick, we end up with a productive process for up to 1 month. So this is the solution to a complex problem. And I think people have been struggling a lot solving that, but the solution is now out there. Whenever somebody has an interest in doing that, we can start straight ahead.
With now 30 days in continuous mode with E. coli, how do you run the DSP? Is that continuous, or is there several batches followed by each other?
No. Project we have been working on with a larger consortium here in India is a fully end-to-end continuous process. So the upstream is continuous, then the primary recovery. In that case, we have our product, so a protein A ligand in the supernatant. So we have to separate the biomass from the supernatant, This is done by continuous dynamic filtration. Then we end up with a clear permeate where our product is located. And this is then captured by a multi-column chromatography approach where we have 4 columns that are run in series. So every time 2 columns are loaded, 1 is eluded and the 4th column is regenerated. And by assembling all these continuous unit operations, we came up with a very intensified process. And all these learnings that we have from changing our mindset in that project to a fully continuous approach, we can inject directly into the classical things we are doing in fat batch mode, because the learnings in process intensification can be also applied to the classical fat batch principle. So how to do it in the most efficient way. Because what is very important in continuous production is that you have a robust setup, because you cannot tolerate failure. So everything has to be very robust and very effective. Otherwise, you cannot master such an endeavor.
What are the main advantages of a continuous process with E. coli? Many of the listeners are familiar with continuous processes with CHO. Are you seeing similar advantages or are they very different?
I think it's quite similar, because what are the main advantages? So you want to produce more with less, meaning less footprint on a facility size, less capital expenditure. So you want to invest in a small facility, not building a 10 cubic meter stainless steel facility. You want to build probably single-use facility that you can fit into a ballroom concept or something like that. And you want to have less operational expenditure. So meaning if it's fully automated and digitalized, you can also save costs on the level of personal costs. I mean, the process that we had been up and running, we had to have one guy available that was looking at the process from a meta perspective, also being able to bling out SMS or something like that if things were going wrong or if there was a deviation. So you can really start to think on a different level. So not having 120 people running around like crazy and trying to have a process that runs with a small footprint and with a low headcount. I think this is what we have to do to innovate, to stay competitive in the race that we are seeing at the moment on a global level. So if we are not willing to innovate, we will become irrelevant. I think this is absolutely the problem we are facing.
Yeah, this is a good point, and I would like to touch upon the innovation a bit later. I just want to ask a question about what can go wrong with an E. coli continuous process, because even with CHO, it's no easy endeavor in certain cases to run a process for 30, 45, or even longer days. What are the main reasons or the main issues you have observed that can go wrong?
The main issues that can go wrong, I mean, the genetic stability firsthand, that is, I think, the major obstacle, as already elaborated on. So this is something we had to learn and also improve stepwise. So genetic stability on the different levels. So genetic stability on the level of the genomes, genetic stability on the level of plasmids that you're using, genetic stability on the level of the expression system, shielding T7 RNA polymerase so that it's not getting hit by mutations, for example, if you're using an orthogonal transcription system like us. And then other issues like sterility, they can be an issue. I mean, we haven't seen a lot because we are very good in working sterile, but this is definitely something, I mean, if you want to have something up and running for more than 2 weeks, it's becoming a problem at some point, especially when you think about single-use equipment where the stuff is validated most of the time just for 10 days or something like that. This is still something with a question mark if you can really run single-use equipment for a long time. What about the leaching issue that people have been addressed already? Is this more pronounced in a microbial process when you use, for example, ammonia? Is there a leaching issue that you don't have in comparison to CHO cells, for example? This is a point to be addressed for the future. But then it's also like the complexity of having unit operations connected to each other continuously also brings some headache at some point, because if something changes, for example, in the upstream, it can have major impact on the downstream. We have solved that on the level of building models around our process, so implementing hybrid or mechanistic models. So one of the nice things I can pull out here is, for example, we are able to determine the product yield every 15 minutes online. So we have an HPLC standing next to our continuous process that is measuring the titer of the product and then injecting that data point into a mechanistic model. And this mechanistic model is counting, for example, the cycles on the multi-column chromatography. So we know how the columns age. And based on this knowledge, we then call the optimal method that is determined by the mechanistic model to purify the protein at the current concentration that we see in the supernatant, for example. So these are like the feedback loops that you can implement to make your life easier. And then, as I also mentioned as a side note before, by automatization, you can also solve a lot of problems on the fly by having readings that tell you, okay, the back pressure is increasing on the filtration device. Let's put out a warning to a guy that is close to the system to have a look at, to be able to counteract upcoming problems early on versus seeing then a burst in a connection or something like that because of an increase in back pressure.
This wraps up part 1 of our conversation. From growth decoupled production to real engineering challenges behind continuous microbial manufacturing, Juergen is giving us a masterclass in rethinking how we scale bioprocesses. And we're just getting started. In part 2, we shift gears into the business side, CDMOs, geopolitics, and the founder's journey. If you are enjoying this episode, leave a review on Apple Podcasts or your favorite platform. It truly helps other scientists like you discover the show. Thank you very much for tuning in today, and I'll see you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.